Okay, we're going to look at section 1.3, which is about experiments for this section. So here we go. So a little bit of terminology to get into before, kind of as we're going through here. So the individuals who are being studied are called experimental units. This may be people, animal, plants, things. Humans don't like to be called experimental units. So oftentimes we are called subjects instead of experimental units. It makes it sound like lab rats. So now we have one of the things we're looking at is the outcome or the response. And this is what's going to be being measured. Oftentimes you think of this as we have an input, which is going to be your things like treatments and stuff like that, which we'll talk here about in a second. And then you have your outputs, which is your like Y values. So it's your response things. So the treatments are the procedures that are being applied. With experiments, oftentimes we may be looking at different treatment levels. It may be treatment versus non-treatment, where there's only kind of two levels as that's processing through. So these are the things that we're looking at. So this first example that they give us is scientists want to determine which of three types of seeds will result in the largest wheat yield. Okay, so the input is going to be the type of seeds, or the treatment will be the type of seeds. The outcome is going to be the amount of wheat yield that we get, okay? So the way that they set this up, since the only thing that we want to be different, okay, is the types of seeds, we want to try and keep everything else as consistent and the same as possible. So we prepare 3 identically sized plots of land with similar soil types. So we're not going to put some of it in, sand and another type in soil and another type in, kind of rocky stuff. Everything's going to be the type of dirt that everything's going into is going to be the same. We're also not going to give one of them, a quarter of an acre, but another type gets a full acre. Like, we're not going to do that kind of stuff. So then we're going to plant each type of seed in a different plot. Okay. So, type A may be in the first one, type B may be in the third one, and type C may be in the second one. It's kind of random. As far as how we feed and water and do that with the plants, it's all going to be the same. So we're going to water and fertilize the exact same way. Same fertilizer, same water, same amount, all that kind of stuff. When we harvest the wheat, we're going to measure the amount that was grown or the yield that we got. And then if one of the types produces substantially more or less wheat than the others, then that one is better or worse than the other two. Okay, so the experimental unit, what this is being done on is the plots of land. The treatment is the type of seed that's being used. And then the outcome is going to be the amount of growth that we have. Now, we do usually have what we call randomized experiments. And this is where the individuals, so the experimental units and the subjects, are randomly assigned to the treatment setups. Okay, so it's not I'm looking at them and going, I want to check something on a new heart medication or something. And I'm going to make sure that all of these people that have heart concerns, I make sure that they get in treatment groups. I'm going to need to have a randomized setup for this so that some people that have heart conditions are in, say, a non-treatment group, and some of them are in treatment. I want people who have a fairly good heart to be in the non-treatment and in the treatment groups. I want to be able to see how this affects, but I need to have that randomization so that I don't have, so that I can have really good data coming out of it, okay? So they're talking about a new effectiveness for a new method of teaching math to elementary school or elementary school children. We've got a random sample of 30 first graders that are taught with the new way. Another set of 30 random first graders are taught with the current way. At the end of 8 weeks, they're given an assessment to test their knowledge. So what they didn't do here is they didn't do Mrs. Smith's class is using the new one, and Mrs. Baker's class is going to go the old way, because Miss Smith may have a different way that she teaches stuff. Miss Baker may have a different way that she kind of does stuff. And so what we want to make sure is that the students are getting the same instruction in the new method if they're in that group, and the same style instruction in the old method if that's the group that they're in. Okay. Now, one of the things that is not under the experiment side, but is actually called an observational study. And with the observational study, this is really kind of what I call people watching, or just data or information gathering. Okay. We're not actually like putting people in and saying, I want you to be in this group and I want you to be in this group and you're going to be taking a treatment or a medication and you're not. So this is one where we may just be asking questions and pulling information. So we've got a study is performed to determine how smoking affects people's health. So we've got a group of smokers and a group of non-smokers. Smokers are observed for several years and they observe the differences in their health outcomes. Okay. We cannot set this up as an experiment in good conscience. Okay. We cannot sit there and take 50 people who don't smoke and say, hey, I need 25 of y'all to start smoking, knowing that there's probably health risk concern with that. At the same time, I can't take 50 smokers and be like, hey, I need 25 of y'all to quit. So this is really just, we're kind of watching them, we're gathering information, but we're not actively asking anybody or putting them in groups to be able to be pulled into one group or the other. So if we take a look at section 1.3 on our worksheet, okay, they're asking us to decide whether it's a randomized experiment or an observational study. So what we've got here is we've got a website developer wants to determine the effectiveness of 2 websites. She provides both designs online. She gets 30 people to look at A, and we've got 30 people who look at B, and then she surveys the 60 people. Okay. This is going to be classed as observational study. Okay. We didn't have anybody trying to, look at one and then go look at the other and decide which one they like better. There wasn't any sort of stuff. This is just, we had 30 people look at each site and then tell us what their opinions were of the site. Okay. So taking a look at number 2, it asks a randomized experiment or an observational study again. Do you think a randomized experiment could be done to study this, or is it going to be observational? So they want to know about a new tire tread. Okay. And they're trying to find out if it's better than an existing with respect to stopping distances and wet conditions. And we have 50 cars. So with this one, this is going to be an experiment. Okay. And the reason being for this is we could do something like 25 cars with the new, 25 cars with the old, okay, or the existing type. And we could put these under different wet conditions, whether it's light, light rain, heavy downpour, things like that. And we can determine, we could run this up, put them on, like the crash dummy type things or whatever, and see if the stopping distances are shorter for the new ones versus the old ones. Is there less skidding? Whatever it is that we're looking for on that one, that we would have, this would be more of an experiment type setup. Okay. So the next thing for this section is why exactly do we want to randomize when we do this? Okay. So in a perfect world, in a perfect setup, the treatment group individuals would not be any different than the other groups, other than what treatments that they get. Okay. So I would have, it's almost like I would have clone copies of each other, that they all had the same health conditions, that I had an individual with, a heart problem and they were slightly overweight and the same type of lifestyle, whatever else, in every group that was being utilized. But we don't have that. Okay. So the best thing we can do is randomize, because when we do the randomization, if we see small differences amongst treatment groups, it's probably only due to chance. Whereas if we see large differences, then that's probably due to the treatments, because we may have kind of some similar stuff going on within the groups as far as the makeup of the groups. Okay. Now, this is talking about a double blind. There is something called a single blind as well. Okay. So we'll start with double blind. In double blind, the investigator or the person taking the information and the subjects who are participating do not know which group they are in. They are, so this is considered double blind. In a single blind scenario, only the subjects don't know. The investigator does know who's at what group. The reason that you would go for this would be something like, let's say, for instance, that I was a nurse or I was I worked in a lab, and it was my job to, you know, I was working in a, let's say, a cancer lab or something like that. And it was my job to, when I got blood work samples and I needed to count, you know, white blood cells and stuff like that in the samples, that what I might do is if I knew patient A was not getting a new medication that was supposed to help with the immune system of cancer patients, I might count theirs twice. That was policy procedure. But if I know patient B is receiving treatment, maybe I count theirs 3 times, or maybe I'm looking a lot closer to make sure I didn't miss anything. I'm not intentionally trying to skew the data or make the data look different, but it might also, because I might put more effort into one counting setup than I do into the other. So double blind would be, I don't know. So if I'm going to double count one, I'm going to double count the other, and I'm going to look at them the exact same. So oftentimes we do take into account double blind scenarios when that's being done. Okay. Now, observational studies are sometimes considered less reliable, especially if you're trying to look at impact of things. So if you're trying to determine whether smoking increases the risk of heart attack, you've got a group of smokers and non-smokers. They're observed for several years. And during that time, we had a higher percentage of smoking group that had heart attacks. Now, there's a little bit of background info in this that they don't tell you, that I would certainly provide, okay, if this was something that we were looking at a little more in depth. They say that one of the problems is that generally men tend to smoke more than women, which means that you are probably going to have more men in your smoking group than ladies, and vice versa. You might have more ladies than men in the non-smoking group. The other concern is that men are actually at a higher risk of heart attack than women are. So because you've got higher heart attack risk men in the smoking group that also has more men, you've kind of got both of those things together. You've got both high risk or high instance scenarios in the same group, and you've got 2 less common things in the same group. So you're seeing this big divide that happens, but that may or may not be actually due to the smoking. It could be that there is some other background stuff that's going on with that we don't, that we can't necessarily pull out and away from that. Okay. Now, the other thing that kind of goes along with that is this thing called confounding. Confounding is when we see some stuff, several different factors that may influence an outcome, and we can't separate it. So the example that I give is that if you are, if we are going to put somebody goes to the doctor and they have high cholesterol, let's say, and the doctor says, look, if you don't get this in check, you're going to seriously, diminish your life. And so the person's like, okay, so the doctor puts them on a medication, but the person also starts exercising. They start eating right. They start, just doing some lifestyle changes. So they go back in 6 months or a year, however long, and the doctor does again, and their cholesterol has dropped by, let's say, 30 points. Okay. Can I say 6 points out of the 30 is because of the medication? Can I say 8 points out of the 30 is because of the diet change? I can't. because we had the diet, the exercise, the lifestyle, and the medications all in there together. And so I don't know how much of that 30 points was contributed to by each factor. Okay. So that's called confounding. And so we can't separate the stuff. Okay. The only way that we would be able to do that would be if the person only went on medication. That's the only thing they changed. They kept their diet the same. They kept their lifestyle the same. They kept their activity level the same. And then when they went back, their cholesterol went down by 8 points. Well, then that 8-point drop was due to the medication. Okay. But if it's all interworked in there together, we can't separate it. Okay. So now in observational studies, taking a look at number 3, we've got heavy drinkers are more at risk for cardiovascular disease than are moderate drinkers. And they ask, is this due to confounding or not due to confounding? This is going to be confounding. It's kind of the same thing with the smoking. You tend to have more men that are heavy drinkers than ladies. And so we've got kind of the same thing. I'm putting a very high frequency thing with another high frequency thing in the same group. And so that's going to be confounding. I'm not going to be able to tell whether or not the heavy drinking actually contributed or was a major factor in that cardiovascular disease. Okay. So now back to our observational studies. We do have in observational studies, it breaks down into 2 subcategories. We've got what are called cohort studies and case control studies. Cohort studies can be broken down a little bit more into prospective, cross-sectional, and retrospective. These all have to do with the time frame of how this information is gathered. Okay, so we're going to take a look at these a little bit closer. So in a cohort study, we've got a group of subjects that are going to be, we're going to be looking at various factors of interest that are associated. Prospective is going to be where you're going to be followed over a long span of time, okay? Or over a span of time. It may be 6 weeks, it may be 6 months, it may be 60 years, okay? One of the famous ones is the Framingham Heart Study. It was started in 1948 with just over 5,200 men and women from the town of Framingham, Massachusetts. Every 2 years, these subjects were given physical exams and lifestyle interviews, and they were studied to discover factors that increase the risk of heart disease. Now, what they don't mention here is that the children, grandchildren, great-grandchildren of these original 5,200 individuals are also included in this Framingham Heart Study. Okay, so they are tracking these people long-term, and they're also looking at offspring and later generations of it to see if there is any sort of generational stuff or hereditary stuff that may also be contributing to this. Okay. Cross-sectional study is one time frame. So it's basically like a snapshot of what's going on. Okay. So they did one where they were looking at the effects of bisphenol A. And this is a chemical found in the linings of food and beverage containers. And so they measured this bisphenol A in 1,455 adults, and they found that people that had higher levels were more likely to have heart disease and diabetes. But the point here is that they did this as a one-time sample grab at that moment. Okay, they didn't come back and check these people like 3 years later. They didn't come back 6 months later or anything like that when they were doing it. Okay. The next or the last type of cohort study is what's called a retrospective. And this is, you're looking at things after something has happened. Okay. So with this one, they got just under 10,000 people who had had gastric bypass surgery in the past 5 to 15 years, along with just under 9,700 people, obese patients, who had not had the bypass surgery, again, in the past 5 to 15 years. When they looked back, they looked to see how many of these roughly 20,000 people were still alive at current. So they're looking at something that occurred in the past and then looking at the outcomes now. And they discovered that the survival rates for surgery patients was higher. So more of the people who had had surgery were still living than people who had not undergone the surgery. So this is, again, we're looking into the past. Oftentimes we may be looking at census data and things like that. In a case control, which is the other type of observational study, they're pulling 2 samples Again, they're pulling from past information, and they're looking at what the interest is. So, for example, with this one, they looked at 201 children who had been diagnosed with brain cancer and 285 that had not been diagnosed with brain cancer. And then they interviewed the children's parents to see what the children's exposure had been to pesticides. They're basically trying to create a link between pesticide exposure and brain cancer. And so again, no one in good conscience is going to subject their child to exposure to pesticides to see if they develop brain cancer. So this is one of those where they're just looking at two setups, and then they can ask questions and see if there was exposure to see if they can link those two things together. So that is the end of section 2.3.